Okay, so thank you everyone for coming. It's very nice to have you here this afternoon for the practical. How many of you have read the practical? Some of you? It's really important to read the practicals in advance. You'll get more out of the practical, you'll understand it more, and you'll do it more quickly, and you'll probably get better marks in the end. So please, I advise you to read the practicals beforehand. Okay, so to give you a little bit of background, my name is Professor Robin Williams. I'm a member of the Centre for Biomedical Sciences here at Royal Holloway. The practical you'll be doing today is based on the research that we do in our lab every day. So to give you a background to that, our lab is funded mostly from industry, but also from the government, and we do work mainly on understanding how drugs and natural bioactive compounds work at a cellular level. So it's surprising that hundreds of drugs used by many people around the world, we know they work because they've gone through clinical trials, but actually how they work, their molecular and cellular mechanisms, remains often unclear. So industry comes to us and says, will you find out how our compounds work? Now, we do this taking an unusual approach. Instead of working on rats and mice, which most neuroscientists and medical researchers do, we use a simple eukaryotic cell, which is an amoeba called Dictyostelium discoideum. So Dictyostelium, as we term it colloquially, or dicty, is a social amoeba. So it's found in the leaf litter of temperate forests around the world, and in that leaf litter, it runs around and eats bacteria. When it's eaten enough, its cells divide in two, and then those run around and eat more bacteria, and they divide, and so on. And that keeps going until the cells run out of food. They then enter into a process of starvation, which triggers a behavioral response where about 100,000 cells will come together and form a small multicellular organism called a fruiting body. And that multicellular organism will have a stalk, which is about 1 millimeter tall, and on top of that a round spore head. And the spores in that will be resistant to starvation and desiccation. So the theory being, an ant or a bunny rabbit will then wander past and touch the fruiting body on the side of its skin or fur, move somewhere else, the spores will germinate where there's more bacteria, and that will allow the cycle to continue. Now, we use this organism to find molecular targets and mechanisms for drugs. One of the most successful areas of research we've done is relating to anti-epileptic treatments, and in particular a compound called valproic acid. This is a very good treatment for epilepsy. It's used around the world for the treatment of epilepsy, but it has some drawbacks in that women who are thinking of having children, if they take valproic acid during their pregnancy, there is a significant chance that the baby, the developing baby, will have birth defects or a lower IQ. So if we can understand how valproic acid works, we could develop alternative treatments based on the mechanism, therapeutic mechanism, but without the side effect of causing teratogenicity, developmental defects during incubation. So the practical you are doing today is to look at Dictyostelium as a model system, and then to look at the effects of 3 compounds on the development process. The first part of the practical, which is the longest, you'll find in your ice buckets Eppendorfs that will be labeled 0 to 5. And they are cells, Dictyostelium cells, that have been grown from 0 to 5 days. And you will use the hemocytometer that you have in front of you to count those cells. and then you'll make a graph of cell number versus days in growth. Also in your ice bucket you will have an Eppendorf with an unknown A, B, C or D, and you will count the cells in that sample, and you'll use your graph to estimate how many days growth those cells have undergone. So that's the first part of the practical. The second part of the practical is you preparing development assays. So in this process you'll calculate how many cells, how much volume of cells you will need to get 1 times 10 to the 6 cells. I suspect you should use day 5, which should have the largest number of cells. So you'll need to work out, based on how many cells are in sample 5, what volume of cells you need to get 1 times 10 to the 6. You'll transfer that volume to a fresh tube. You'll spin it down with a microfuge on the side or over in the corner for a couple of minutes. Please make sure you balance the microfuge. So there are tubes on opposite side of the microfuge with the same volume. You'll pour off the supernatant just down the sink is fine. You'll add some phosphate buffer, KK2, that's in your ice box with a red cap. You'll resuspend it, and then you will prepare a development assay, and I'll show you later on in the practical how to do that. So then you'll give that to myself or our expert demonstrator, Sonia Shinmar, who's here. She's a fantastic PhD student in the lab. And we will incubate them overnight, and you can come tomorrow afternoon to have a look at the development effects. Each of you will have in your ice buckets a compound, either KK2, which is a control, or valproic acid, VPA, or ethanol, or retinoic acid, which is another teratogenic compound. So you will add those chemicals to your development assay, and you'll come back tomorrow and you can see what happens. But because that takes 24 hours, the third part of the practical we have prepared already for you, and this is on the side of the lab here, and you can come and have a look at what normal development looks like and what development after incubation with these compounds, what happens. So that's the practical in essence, and in a second I will show you how to use the hemastometer.